Hello everyone and welcome to our channel MooTechie. A special welcome to all the Dairy Technologists and students of food science and related courses. We're glad you're here to learn with us. Today, we begin our journey into the fascinating world of food microbiology and ITS Part 1. Our focus will be on the foodborne microorganisms that are so crucial to your field. Let's start by understanding the major groups of microbes important in foods. The microbial groups important in foods consist of 7 aspects and types of bacteria, yeasts, molds, and viruses. It's interesting to note that while some algae and protozoa, and even some worms like nematodes are important in foods, they are not included among these core microbial groups. Some of the protozoa and worms are associated with health hazards. Several algae are also associated with health hazards and with bioprocessing, where they serve as sources of vitamins and single-cell proteins. But our main players are bacteria, yeasts, molds, and viruses. They are important in food for 3 key reasons: their ability to cause foodborne diseases, both spoilage, their role in food spoilage, and their use in producing foods and food ingredients. Now let's talk about their roles in more detail. Many bacterial species and some molds and viruses are able to cause foodborne diseases. Notice that yeasts are not typically capable of this. When it comes to spoilage, most bacteria, molds, and yeasts can potentially cause food spoilage because they are able to grow in foods. Viruses, however, cannot grow in foods. Moving on, we will see the positive side of these microbes. Several species of bacteria, molds, and yeasts are considered safe or food grade or both. These are the ones used to produce fermented foods and food ingredients that we all enjoy. Among the 4 major groups, bacteria constitute the largest group. Because of their ubiquitous presence and rapid growth rate, even under conditions where yeasts and molds cannot grow, they are considered the most important in both food spoilage and foodborne diseases. Exploring classification and nomenclature. Next, we explore the classification of microorganisms. Living cellular organisms, on the basis of phylogenetic and evolutionary relationships, were originally grouped in 5 kingdoms. In this system, bacteria belong to prokaryotes, which means before nucleus. The eukaryotic molds and yeasts, which have a true nucleus, were grouped under the kingdom fungi. Viruses are not considered as living cells and are not included in this biological classification system. For the classification of yeasts, molds, and bacteria, several ranks are used after the kingdom. These ranks are divisions, classes, orders, families, genera, that's the plural for genus and species. The basic taxonomic group is the species. Several species with similar characteristics form a genus. In food microbiology, ranks above species, genus, and family are seldom used. Among bacteria, a species is regarded as a collection of strains having many common features. Now let's look at the concept of a strain. A strain is the descendant of a single colony, which originates from a single cell. Among the strains in a species, one is assigned as the type strain. This type strain is used as a reference strain when comparing the characteristics of an unknown isolate. Now, we come to nomenclature. The basic taxonomic group in bacteria, yeasts, and molds is the species, and each species is given a name. The name has 2 parts, making it a binomial name. The first part is the genus name and the second part is the specific epithet, vile, which acts like an adjective. Both parts are Latinized. When written, they are italicized or underlined. The first letter of the genus is always written in a capital letter. For example, we have Saccharomyces cerevisiae, Penicillium roquefortii, and Lactobacillus acidophilus. A bacterial species can be divided into several subspecies, abbreviated as subspe, or SSP. This happens if the members show minor but consistent differences in characteristics. Under such conditions, a trinomial epithet is used. An example is Lactococcus lactis SSP, Lactis or Lactococcus lactis SSP, Cremoris. In some instances, ranks below subspecies are used to differentiate strains recognized by specific characters. For example, we have serovar, based on antigenic reaction, biovar for strains producing a specific metabolite, and phagovar for strains sensitive to a specific phage. Such ranks have no formal taxonomic importance, but can be practically very useful. For instance, Lactococcus lactis SSP, Lactis, biovar diacetyl. Lactis is a strain that produces diacetyl. Diacetyl is an important flavor compound in some fermented dairy products. Each strain of a species should be identified with a specific strain number. This number can be alphabetic or numeric or a mixture of both. An example is Pediococcus acidilactici, LB 923. At the family level, bacterial names are used as plural adjectives in feminine gender and agree with the suffix aci. A well-known example is Enterobacteriaceae. The species and strains in a genus can be represented collectively. This is done either by using SPP, after the genus name, for example Lactobacillus spp, or we can use the plural forms of the genus. For example, we say Lactobacilli for Lactobacillus, Lactococci for Lactococcus, Leuconostox for Leuconostoc, or Salmonella for Salmonella. The scientific names of bacteria are given according to the specifications of the International Code of Nomenclature of Bacteria. The International Committee on Systematic Bacteriology of the International Union of Microbiological Association examines the validity of each name. This committee then publishes the approved lists of bacterial names from time to time. A new name for a species or genus must be published in the International Journal of Systematic Bacteriology before it is judged for inclusion in the approved list. Any change in a name, whether genus or species, has to be approved by this committee. Morphology and structure of microorganisms. In the following section, we discuss the morphology and structure of microorganisms in foods. Both yeasts and molds are eukaryotic. But yeasts are unicellular, whereas molds are multicellular. Eukaryotic cells are generally much larger than prokaryotic cells. Their size ranges from 20 to 100 micrometers, while prokaryotic cells are typically 1 to 10 micrometers. Eukaryotic cells have rigid cell walls and thin plasma membranes. The cell wall does not have mucopeptide. It is rigid and is composed of carbohydrates. The plasma membrane contains sterol. The cytoplasm is mobile, a phenomenon called streaming gaia, and contains membrane-bound organelles like mitochondria and vacuoles. Ribosomes are the ATs type and are attached to the endoplasmic reticulum. The DNA is linear, organized into chromosomes, contains histones, and is enclosed in a nuclear membrane. Cell division is by mitosis tria, which is asexual reproduction. When sexual reproduction occurs, it is by meiosis. Molds, eukaryotic, multicellular. Now let's describe molds in more detail. Molds are non-motile, filamentous, and branched. The cell wall is composed of cellulose, chitin, or both. A mold body, called a thallus, is composed of large numbers of filaments called hyphae. An aggregate of hyphae is called a mycelium. A hypha can be non-septate, meaning without divisions, septate uninucleate, with divisions and one nucleus per section, or septate multinucleate, unio, with divisions and multiple nuclei. A hypha can be vegetative for growth or reproductive. The reproductive hypha usually extends into the air and forms exospores. These spores can be free, called conidia, or in a sac, called a sporangium. The shape, size, and color of spores are used for taxonomic classification. The diagram shows the structure of a mold. We see hyphae at the base. From these, an aerial hypha grows up, forming a conidiophone at the top. This conidiophone produces conidia, which are the spores. These spores can then germinate and form new subsurface hyphae, continuing the life cycle. Yeasts, eukaryotic, unicellular. Now let's look at yeasts. Yeasts are widely distributed in nature. The cells are oval, spherical, or elongated, about 5 to 30 micrometers by 2 to 10 micrometers in size. They are non-motile. The cell wall contains polysaccharides, known as glycans, as well as proteins and lipids. The wall can have scars, indicating the sites of budding, which is how they reproduce. The plasma membrane is located beneath the wall. The cytoplasm has a finely granular appearance due to the presence of ribosomes and organelles. The nucleus is well defined with a nuclear membrane. Bacterial cells, prokaryotic, unicellular. Coming up, we examine bacterial cells. Bacteria are unicellular. Most are about 0.5 to 1.0 micrometers by 2.0 to 10 micrometers in size. They have three basic morphological forms: spherical, called cocci, rod-shaped, called bacilli, and curved, like a comma. They can form associations such as clusters, chains of two or more cells, or tetrads. They can be motile or non-motile. Cytoplasmic materials are enclosed in a rigid wall on the surface and a membrane beneath the wall. Nutrients in molecular and ionic form are transported from the environment through the membrane by several specific mechanisms. The membrane also contains energy-generating components. It also forms intrusions in the cytoplasm called mesosomes. The cytoplasmic material is immobile and does not contain organelles enclosed in a separate membrane. The ribosomes in bacteria are the 70S type and are dispersed in the cytoplasm. The genetic materials, which include structural DNA and plasmid DNA, are circular. They are not enclosed in a nuclear membrane and do not contain basic proteins such as histones. Both gene transfer and genetic recombination occur in bacteria, but they do not involve gamete or zygote formation. Cell division is by binary fission. Prokaryotic cells can also have flagella for movement, capsules for protection, surface layer proteins, and pili for specific functions like attachment. Some bacteria also form endospores, with one endospore per cell. This is a dormant, highly resistant structure. On the basis of Gram-stain behavior, bacterial cells are grouped as gram-negative or gram-positive. Gram-negative cell wall. Gram-negative cells have a complex cell wall containing an outer membrane and a middle membrane. The outer membrane is composed of lipopolysaccharides (LPS), lipoprotein, and phospholipids. The outer membrane has limited transport and barrier functions. The resistance of gram-negative bacteria to many enzymes like lysozyme, to hydrophobic molecules like SDS and bile salts, and to antibiotics like penicillin is due to the barrier property of this outer membrane. LPS molecules also have antigenic properties. Beneath the outer membrane is the middle membrane. It is composed of a thin layer of peptidoglycan or mucopeptide embedded in the periplasmic materials that contain several types of proteins. Beneath the periplasmic materials is the plasma or inner membrane. This is composed of a phospholipid bilayer in which many types of proteins are embedded. Gram-positive cell wall. Now let's look at gram-positive cells. Gram-positive cells have a thick cell wall composed of several layers of mucopeptide. This is responsible for the thick, rigid structure. The wall also contains two types of teichoic acids. Some species also have a layer over the cell surface, called surface layer protein or SLP. The wall teichoic acid molecules are linked to the mucopeptide layers. The lipoteichoic acid molecules are linked to both the mucopeptide and the cytoplasmic membrane. Teichoic acids are negatively charged because of their phosphate groups. This may bind to or regulate the movement of cationic molecules in and out of the cell. Teichoic acids also have antigenic properties and can be used to identify gram-positive bacteria serologically. Because of the complexity in the chemical composition of the cell wall, gram-positive bacteria are considered to have evolved before gram-negative bacteria. We now have a diagram of a generalized bacterial cell. The main body is the cytoplasm. Within it, we see the chromosome and ribosomes. Also in the cytoplasm is an endospore. On the outside, the cell is enclosed by a cell membrane and a cell wall. Additional structures attached to the cell include a flagellum, pili, and a capsule surrounding the entire cell. Following this, we notice a comparative diagram of cell walls. On the left is the gram-positive cell wall. From the inside out we have the cytoplasm, the cytoplasmic membrane, a thick layer labeled peptidoglycan A, and then lipoteichoic acid, or LTA, extending through it. Proteins and phospholipids are part of the membrane structure. On the right is the gram-negative cell wall. From the inside out we have the cytoplasm, the inner membrane Yeasts, which is a phospholipid bilayer. Then we have the periplasmic space, which contains a thin layer of peptidoglycan. Outside this is the outer membrane. This outer membrane contains lipopolysaccharides, LPS, lipoprotein, and porins, which are proteins that form channels. Viruses, non-cellular entities. Now, we discuss viruses. Viruses are regarded as non-cellular entities. Bacterial viruses, called bacteriophages, are important in food microbiology and are widely distributed in nature. They are composed of nucleic acids, either DNA or RNA, and several proteins. The proteins form the head, which surrounds the nucleic acid, and the tail. A bacteriophage attaches itself to the surface of a host bacterial cell and inoculates its nucleic acid into the host cell. Subsequently, many new phages form inside the host cell and are released outside following lysis or bursting of the cell. Several pathogenic viruses have been identified as causing foodborne diseases in humans. However, because they are difficult to detect in foods, the involvement of other pathogenic viruses in foodborne diseases is not correctly known. The two most important viruses implicated in foodborne outbreaks are hepatitis A and Norwalk-like viruses. Both are single-stranded RNA viruses. Hepatitis A is a small naked polyhedral enteric virus about 30 nanometers in diameter. The RNA strand is enclosed in a capsid. Important microorganisms in food. Moving on, we look at the important microorganisms in food, starting with important mold genera. Molds are important in food because they can grow even in conditions in which many bacteria cannot grow. These conditions include low pH, low water activity, written as AW, and high osmotic pressure. Many types of molds are found in foods. They are important spoilage microorganisms. Many strains also produce mycotoxins and have been implicated in foodborne intoxication. On a positive note, many are used in food bioprocessing. Many are used to produce food additives and enzymes. Key mold genera. Aspergillus, widely distributed. Septate hyphae. Produce black-colored asexual spores on conidia. Many are xerophilic. Spoil grains, jams, cured ham, nuts, fruits, and vegetables. Some produce mycotoxins, e.g., Aspergillus flavus produces aflatoxin. Used in food processing, e.g., ants asp. Orizae forsake asp. Niger for citric acid and enzymes. Alternaria septate. Form dark-colored spores on conidia. Cause rot in tomatoes and rancid flavor in dairy products. Some strains produce mycotoxins. Example: Alternaria tenuis, Fusarium, associated with rot in citrus fruits, potatoes, and grains. Form cottony growth and septate, sickle-shaped conidia. Example: Fusarium solani. Geotrichum, septate, form rectangular arthrospores. Grow as a yeast-like cottony, creamy colony. Establishes easily in equipment, often on dairy products, dairy mold. Example: Geotrichum candidum, mucor. Widely distributed. Non-septate hyphae, produce sporangiophores. Produce cottony colonies. Some used in food fermentation, and as an enzyme source. Cause spoilage of vegetables. Example: Mucorrhuxi, penicillium, widely distributed. Septate hyphae, form conidiophores on a blue-green brush-like conidia head. Some species used in food production, e.g., Penicillium roquefortii and Tainpen, Camembert ** in cheese. Many cause fungal rot in fruit/vegetables, spoilage of grains, breads, and meat. Some strains produce mycotoxins, e.g., ochratoxin A. Rhizopus hyphae are aseptate and form sporangiophores in a sporangium. Cause spoilage of many fruits and vegetables. Rhizopus stolonifer is the common black bread mold. Key yeast genera. Yeasts are important in food because of their ability to cause spoilage. Many are also used in food bioprocessing and to produce food additives. Saccharomyces, round, oval, or elongated cells. Most important genus. Saccharomyces cerevisiae variants are used in baking for leavening bread and in alcoholic fermentation. Also cause spoilage producing alcohol and CO2. Pichia, oval to cylindrical cells. Form pellicles in beer, wine, and brine to cause spoilage. Some used in oriental food fermentation. Example: Pichia membranae efficiens. Rhodotorula, pigment-forming yeasts, causing discoloration of foods such as meat, fish, and sauerkraut. Example: Rhodotorula glutenis. Torulopsis, spherical to oval cells. Cause spoilage of milk, because they can ferment lactose. Spoil fruit juice concentrates and acid foods. Example: Torulopsis versatilis. Candida. Many species spoil foods with high acid, salt, and sugar, and form pellicles on liquid surfaces. Some cause rancidity in butter and dairy products. Example: Candida lipolyticum. Zygosaccharomyces cause spoilage of high acid foods, such as sauces, ketchups, pickles, mustards, mayonnaise, and salad dressings. Example: Zygosaccharomyces bailey. Important viruses in food. Viruses are important in food for three main reasons. 1. Cause enteric disease and foodborne diseases, e.g., hepatitis A and Norwalk-like viruses. Others include poliovirus, echovirus, and Coxsackie virus. 2. Bacteriophages can be used to identify some pathogens, e.g., Salmonella species and Staphylococcus aureus strains, based on their sensitivity. 3. Bacteriophages are used to transfer genetic traits, transduction, in some bacterial species, e.g., Escherichia coli or Lactococcus lactis. 4. Cause fermentation failure. Phages can infect and destroy lactic acid bacteria starter cultures, causing the fermentation process to fail. Phages have been isolated for Lactococcus, Streptococcus, Leuconostoc, and Lactobacillus. No bacteriophage of Pediococcus is yet known. Important bacterial genera. Bacterial classification is changing rapidly. The 9th edition of Burge's Manual of Determinative Bacteriology, 1993, lists more than 560 genera. New genera continue to be created, such as Lactococcus, former N group or dairy Streptococcus, and Carnobacterium, some former Lactobacillus species. Bacterial shapes. We have three main categories. 1. Spheres, Streptococci, chain of circles, Streptococcus pyogenes. Diplococci, pairs of circles, Streptococcus pneumoniae. Tetrad, square of 4 cells. Staphylococci, irregular cluster of circles, Staphylococcus aureus. Sarcina, cube of 8 cells, Sarcina ventriculi. 2. RODS, bacilli, chain of bacilli. Bacillus anthracis, flagellate rods, Salmonella typhi. Spore former, rod with a central endospore, Clostridium botulinum. 3. Spirals, Vibrios, curved rods, Vibrio cholerae. Spirilla, rigid spirals, Helicobacter pylori. Spirochaetes, flexible spirals, Treponema pallidum. Finally, we have an image titled Anthrax bacterium, Bacillus anthracis. It shows a detailed diagram of a rod-shaped bacterium with a large capsule surrounding it, visible as a clear halo around the purple-stained rods, capsule staining. Inside the cell, the components are labeled DVA, likely DNA, cell membrane, cell wall, and the outer capsule. An endospore is also detailed, containing spore DNA and ribosomes, surrounded by the spore wall, spore membrane, and a layer called the cortex. Thank you for joining this comprehensive overview of foodborne microorganisms. We covered the major groups: bacteria, yeasts, molds, and viruses, their structures, how they are classified, and key genera important in food spoilage, disease, and production. Remember, understanding these microbes is the first step in controlling them and harnessing their power in dairy and food technology. We hope this was helpful. Thank you. See you in the next class.